Okay, so now you know something about me. I want to find out a little bit about you all, okay? How many of you are studying biology? Wow, what kind of place? How many of you are studying chemistry or chemistry majors? Don't be shy. Oh, great. Okay. Let's see. How many are studying some aspect of environmental science and environmental studies? One back there? Okay. Two. Okay. All right. Okay. Among those of you, since most of you are in biology, give me a sense of how many are interested in the broader biomedical field? Biomedical field? Okay. Some of you like pre-med or pre-dent. Oh, okay. Yeah, biomedical field. Okay. If you're interested in being pre-med, pre-dent. So many of you rose your hand. Give me an idea of what aspect of biology those of you who are not interested in pre-med are interested in. Can you give me some idea? Education. Okay, good. Anybody else? Okay, that's fine. I just wanted to get a sense of your background. Okay, so I am just going to start the slides. The pictures down at the bottom are ones that we will, most of them, I don't think I'm talking about this one, but we'll be talking about these, okay, in terms of how it fits into the story that I'm telling you, that I'll be telling you. Okay, let's now how do I? Okay, there we go. Is it that I cannot? Okay, there we go. This is just an overview to give you a sense. I'm going to talk a little bit about the path that got me here. Because I was a microbiology, like many of you were, and discuss how the right timing can help a lot. And then I'm going to tell you about what I learned from chemists, environmental chemists, and what I learned from environmental engineers. And how that sort of helped me end up where I am with regard to the classical microbiology training that I had, and then working and mucking about in the months of New Jersey and New York. And then just a closing message to the students. Okay, so that's the idea. So you get a sense of how all this happened, and you know the list of things that Dr. Healy was mentioning is because I've been around a long time. So if you've been around a long time, you know, you end up being assigned all sorts of things that you have to do. And so it'll give you a sense of how that all, hopefully, all that comes together. Okay. Okay, the path that got me here. As I mentioned, last century, now not many people can say that, but last century, I was a microbiology major as an undergraduate at this was Cornell University. This is the way it is, sort of like 10 months out of the year, but that's not true. And I studied a lot of microbiology that at that time, I'm old enough to be of your parents or your grandparents' generation. Okay, so we're talking about a lot of them. At that time, the microbiology was highly biomedically oriented because that's where the subject was very important to disease and pathogens and such. So there wasn't much attention, there wasn't any attention with regard to its role in the environment, which it is now. Okay, it's changed. I learned chemistry from a professor called Werner Schumm. And the chemistry I learned was as a graduate student, because as an undergraduate, those of you who are biology and chemistry majors are not interested in the year of chemistry, or the year of organic, the year of, let's see, the year of analytical and people. And I had 4 years of chemistry, none of which I really enjoyed and chemistry, until I took a graduate course from Professor Schoen, who is considered the father of applied chemistry. He wrote the first book, and he focuses on the chemistry of natural waters and wastewater treatment, but focusing on natural waters. And through that, he also recognizes that there's a lot of microbiology that occurs in these natural waters to keep the system working, to keep the oceans and the lakes and the rivers all working like the way they should. So there's microbiology in the chemistry that he taught. And that then I got really interested in chemistry, because it became very clear that microbes, from his point of view, they're just carrying out chemistry. They're carrying out oxidation reduction reactions, just like you can write in terms of the chemical reaction, oxidation reduction. But that's what they're carrying out, oxidation reduction reactions. Then I learned engineering application from Professor Perry Hardy, who was director of the program where I had the first job. And my first position was in an civil and environmental engineering department. So here I am trained as a microbiologist. Why? Well, because this department also had a vision that engineering, environmental engineering, civil engineering, is very much linked with microbiology, especially in terms of wastewater treatment. As you know anything about wastewater treatment, which has worked for the last 100+ years in the US to keep our waters clean and to prevent typhoid and cholera and all that from just going into the population because of wastewater treatment that the engineers dealt with. And that was microbiology. In the wastewater treatment system, it's all the microbes that are getting rid of the things that you don't want in the wastewater. And then this was my professor that I did my PhD with, Ralph Mitchell. And what was interesting with hindsight is that Professor Mitchell was interested in microbiology in many different sectors. He was trained as a plant pathologist, but then he was doing microbiology on surfaces, all sorts of different surfaces. And when he retired, he was doing microbiology of degrading of artwork that degrades, in museums, because there's fungal issues on many old manuscripts and beautiful paintings you see in museums. There are entire departments that take care of them in the large museums. So those are the people who influenced me to this point, where even though I started as a microbiologist studying biomedical microorganisms, I'm now mucking about in the environment. Okay. The New Jersey muds and the New York Harbor muds. So I just wanted to point that out, that there's an evolution, and that in every place I learned something that applied to my understanding. And that understanding became more and more focused on understanding what happens in the environment and the role that microorganisms have. Okay, the right timing can help a lot. This is a picture of Prince William Sound in Alaska. Beautiful, pristine environment, which in 1989, because but your parents would remember, your grandparents would remember, the Exxon Valdez oil tanker that's battered down and spilled 18 million gallons of crude oil in this beautiful, pristine wilderness. It became worldwide news because of the large volume, but also because it sort of decimated this ecosystem that was just so beautifully clean and unharmed and just totally destroyed. Now, the beaches, yeah. Am I, okay. Then I can't, I can't, I don't think I can do it this way. Okay, that's better. Okay, this is better for me too. Okay, so the beaches in Alaska, it's nothing like the Jersey Shore. Okay, it's not sandy, beautiful, white beaches. There's stony beaches, and the oil would then cover these beaches, and because they're stony beaches and rocky and very heavily and everything, all this oil would go into the interstices and into the crevices. And you can see what, if you picked up contaminated beach material, this is what it looked like. Okay, so now I have to go to the next one. The impact. The impact was phenomenal when you think about it. Valdez is up here. And this is Prince William Sound, this region right here. And so after the tanker ran aground and spilled, slowly within weeks, days and weeks, the oil then, due to the flow of the water and the currents, just contaminated the shoreline all the way down. And this is essentially the entire bottom part of Alaska, if you want to think of it in terms of the way we look at Alaska on a regular map. 1,300 miles of shoreline were contaminated with all this sticky, gooey oil. And that's the distance. Think about this. This is the distance from New York to Florida. So you can imagine if the entire coast from New York to Florida was contaminated with an oil tanker losing its tank full of oil. And the destruction was horrendous. You can see the numbers. Hundreds of thousands of seabirds, sea otters, harbor seals, bald eagles, orca whales, billions of salmon and herring, and the fishing industry in Alaska was just totally decimated for a number of years. The cost of cleanup, Exxon paid more than $3.8 billion to clean up the habitat restoration and personal damages for people who lost their livelihoods. There was an additional $3 billion estimated to have cost EPA and the state of Alaska. If we look at, yeah.
Oh, be helpful. Do you have a USB?
Oh, no, I don't have
No?
that.
Okay.
That's okay. Okay, let me do it from here. Now, if you can take a look at this, this set of pictures, this shows the contamination along the shoreline, okay, in this map. But it also shows how they cleaned it up with booms on the water. They would just collect the oil and cart it away. You can see what the beaches look like. This is the way the contaminated beach would look. And you can see how they were trying to clean it up. Now, a couple of things should be kept in mind. One is that they're still using these kind of techniques. Same kind of thing if there's a more recent spill, you still collect them with these booms. Now, in Alaska, because of the terrain and because of the liability that Exxon had, they hired hundreds and hundreds of people to wash the beaches with hot water. You can imagine the beaches, all these beaches. I don't know if they washed every single beach, but they washed the beaches with hot water to just wash the oil off the beaches back into the ocean. And so that took a long, long time. Okay, so in regard to recent spill, you may remember, although you may have been very young, like 4 or 5 years old or something like that, but this was the the BP oil spill, the Deepwater Horizon drilling rig that exploded in the Gulf of Mexico. And they couldn't plug this up for 4 months in terms of the wellhead, which just broke off. And so oil was just flowing out of the ground, the subsurface. It was in the water, out of the subsurface for months and months. But again, physical labor was required to clean up the beaches. Here, they're just digging up the contaminated soils and sand and on the beaches and the oil and hauling it away. Okay, so this is the kind of beaches that it affected. And then this is actually a way of getting rid of some of the oil on the surface, is to just burn it off. Okay, so these are very expensive, intensive, and not very efficient ways of cleaning. So to just fast forward, the EPA had microbiologists, many of whom I knew from just our common interest in work, who understood that oil is a natural product and it is biodegradable. And there are organisms in the environment, and there have been organisms studied for a long time in laboratories that can use oil as a food source. And we'll talk more about that. So they decided, okay, let's see if we can't use these this capability that these organisms have to clean up some of the contamination. And what I'm showing here is a photo from the Environmental Science and Technology 1991 publication. What they did was they used a whole different series of these oleophilic fertilizers. So this is fertilizer which has sort of been embedded in waxy, sort of like peanut butter consistency, so that it doesn't just wash away. If you just sprinkle, this is like fertilizer you would use on your garden, but if you just sprayed on the nitrogen, phosphorus as a solution, it'd just be washed out by the tide, right? So this is the tree line, and this is the water line. And so they tested these in different quadrants along some of the contaminated beaches. I know Atlas and Bartha, Ron Atlas and Richard Bartha pioneered one of the formulations, which was found to be very successful because Professor Bartha was a Rutgers professor in the microbiology department, and Ron Atlas was his graduate student. They had worked on this even before this contamination. They worked on oil biodegradation. So what you can see is that after a couple of weeks, okay, here are two people to give you a size of the a sense of the size of this, the size of these quadrants. You can see that the beach here, which had been applied with this oleophilic fertilizer, appeared much cleaner than near the tree line or down here where no fertilizer was applied. So it was clear that the biodegradation of oil was limited by nitrogen and phosphorus, by just fertilizer. And why? Because hydrocarbons are carbon and hydrogen. A lot of carbon. If it's carbon, then if a microbe can use it as food, can turn it into energy, and then reproduce, you will find a microbe to do that. Okay, and so they're in the environment. It's not a new invention. It's not a new formulation that no microbe has ever seen. It's the hydrocarbons and petroleum are natural products. At that time, the EPA director, administrator was a man named William Riley at the time. And he was so impressed with the results as he flew over these contaminated shorelines and looked at the results. He then supported and pushed the momentum for research supporting bioremediation of contaminants in the environment. So that was from the early 1990s for about 20-25 years, where there was a great interest and a lot of work that we did on biodegradation, as well as many other labs. Okay, so it became something that there was support for. How can we use microbes to decontaminate the environment in a way that is less harmful to the environment than what? Than digging it up and hauling it away, but then you have to put it somewhere. You have to find a place to put it. So that was being a good timing. Timing can make a difference. It just happened to be during the time when I had the opportunity to look at these kinds of issues. So good timing is important. Okay, so here are the structures of some of these compounds, which are probably quite familiar to you. BTXs, benzene, toluenes, and xylenes. It's benzene with some methyl groups attached to it in different positions. It's the main component of gasoline. Naphthalene, phenanthrene, polycyclic aromatic hydrocarbons. They're found in petroleum, and they're found in the crude petroleum and the kind of petroleum that contaminated the beaches that we saw pictures of. PCBs, polychlorinated biphenyls. This has been much interest to not just microbiologists, but to GE, which had a huge plant near Albany, New York, and had disposed of the PCBs products that they had worked with, that they had produced into the Hudson River. At that time, the disposal was not illegal. It's not like they illegally disposed of it, but it was later that it was determined that these PCBs were very, very slow to degrade, just like what is now the PFASs, the fluorinated carbon compounds are even more difficult to degrade biologically, microbiologically. But people worked on the fluorinated compounds and found that there is biodegradation that can occur for the PCBs and also for the solvents, the PCE, perchloroethylene, TCE, trichloroethylene, DCE, dichloroethylene. These are solvents which are used to just clean machine parts. The military used lots of it to clean its automobiles and all sorts of materials that they needed to keep clean. And then dry cleaners, the old dry cleaner solutions were perchloroethylene and trichloroethylene. And you have that funny dry cleaner smell, and that's what you're smelling. And then that's chlorine carbon bond too, which we now know can be broken. Mostly these things are occurring in the presence of oxygen. In the absence of oxygen, then that's a whole nother story. Okay? How do these organisms degrade it in the absence of oxygen? And I'd like to just sort of make a case for, in the environment, the anoxic world. Okay, the toxic world is very thin in terms of the global structure. The anoxic world is much, much deeper. And of course, it was easier and more convenient to study the aerobic world from a microbiology point of view in the environment. And the anoxic world, unseen, out of sight, out of mind, just became less studied. Okay, so then the question that I got involved in, because the engineers were interested in this, is can these kinds of compounds be degraded in the absence of oxygen if it's a subsurface kind of environment? Because groundwater is in the subsurface. All sorts of other aquifers are in the subsurface. Sediments are especially very active, and very rich sediments are very readily deoxygenated in the thin surface that's exposed to oxygen. And then there's this whole anaerobic region or anoxic region. So that becomes the question. Okay, so what did I learn from the environmental chemists? So these are some of the lessons. I learned that microbes are carrying out chemistry, and they're really the best chemists in the world. I learned that if there is food, if there's carbon available and a source of energy for the organism, like oxygen, they respire oxygen, but I'm going to make the case that they can respire other compounds too in place of oxygen. So if they have a source of food and a source of energy, then you can find a microbe to use it. You will find a microbe to use it. They are the only organisms in our biosphere that can eat, so to speak, petroleum constituents and breathe something other than oxygen. So something else can substitute for it, and we'll talk about that. And microbes are the mediators of essential oxidation reduction reactions that are environmentally important. So chemistry then became microbiology for me, and I began to appreciate the importance of chemistry for the microbiology that I was interested in. Okay, so here's some of the chemistry, just a very short run through. Now, we all know that glucose can be converted to CO2 and water, and there's energy that's produced. And there's enough energy to produce to make new cells. Okay, that's how they make a living, organisms and any of the cells in our body. We make a living by using glucose, expelling CO2, and using that energy to make new cells. Same thing with us. So benzene, toluene, and xylene, as examples, are also in this class where they can be oxidized to CO2 and water and cells. So how do we get energy? We get energy by the electron accepting reactions, by oxygen going to water, okay? And that energy can be measured by this delta G, the energy of transformation in kilocalories per 2 electron transfers, electron pair. Okay, so the point is that oxygen is commonly available in the environment under aerobic conditions. Okay? Now, there are other commonly available compounds in the environment that microbes can use instead of oxygen. Nitrate. Nitrate can be reduced to nitrogen gas. It provides almost as much energy as oxygen. And there are many microbes that can use nitrogen instead of oxygen. So they don't have to be right at the surface. And nitrate is readily available in the environment. Iron. Iron 3 is reduced to iron 2. It does provide energy. And it is widely available in the environment. These other, let's see, what is it? This is chromium and this is arsenic. Those are not widely available in the environment. So let's stick to the environmentally relevant ones. Sulfate. Sulfate is widely available in the environment. It gets reduced to sulfide, and that provides energy, but it's very little energy. So the sulfate reducing organisms in the deep subsurface at very low redox conditions have to work very hard. They do get energy from it, but you can see that they have to process this reaction many more times to get this amount of energy out of it. Okay, so they have to work harder, but it's widely available in the environment. And then carbonate. Carbonate is the source of carbon for methane. Methane, we know, is a greenhouse gas and is also the product of anaerobic processes. And that's why the carbonate is widely available in the environment. And again, it provides very little energy compared to oxygen, but it still provides energy for the organism and is widely available in the environment. So these compounds are all widely available as a substitute for oxygen, these other compounds, as a substitute for oxygen in an anoxic environment. And so that's the argument that I'm making. And here, let me show you work that is classic now. This is an aerobic naphthalene degradation pathway. And it's the thing I want to point out is that the biochemists who worked on this, which was more than 50 years ago, just elegantly, without the kind of instrumentation and the techniques that we have today, could work out these incredibly elaborate pathways and help us understand what's happening. Here's naphthalene. Now, naphthalene gets activated so that it can be degraded. These are very stable chemical structures with these oxygenase enzymes. This is a dioxygenase enzyme, so 2 oxygens are needed. And these oxygens are taken from the atmosphere, So these oxygenases can take oxygen from the atmosphere, put it onto the molecule to help destabilize it so that it can be further degraded. here's another dioxygenase reaction, which can destabilize the ring and allow it to then be further degraded. So what this tells us back then, before we understood the anaerobic processes, is that for these aerobic hydrocarbon degraders, they must take molecular oxygen from the atmosphere to work. So people say, anaerobically, this kind of stuff is not going to happen. No, it doesn't happen in the same way, but it happens. It happens in a different way. There's different chemistry involved. Okay, so this is where Professor Healy was talking about sampling in and around New York. Okay, you can see Manhattan. You can see the greater harbor, New York Harbor. You can see Staten Island, and here's the Arthur Kill, which is an intertidal waterway between Staten Island and New Jersey. And I was at NYU Medical Center for a long time, and this is where we sampled. We sampled in the East River, we sampled sediment from the East River, we sampled sediment from the Arthur Kill. Now, why did we go to the Arthur Kill? Because that's a wonderfully contaminated intertidal waterway. And because it's so well contaminated for so long, we figured, okay, if there's any organisms that can degrade these nasty compounds, maybe we'll find them in the Arthur Kill. On the New York side of the Arthur Kill was the largest landfill in the world, the Fresh Kills landfill. On the New Jersey side is Elizabeth, New Jersey. You know where those refineries are when you go up and down the turnpike, right? And so there are spills from the refineries going into the Arthur Kill. And this landfill is very old. It was very big. It's closed now. And then the tide would come in and would go into the landfill, and the tide would come out and it would take all the leachate from the landfill and deposit it into the sediment and the waterways of the Arthur Kill. So the sediment, we would take sediment from there with these drop cores. You can see how dark it is, and it has a consistency of black peanut butter, but it's anoxic because it's so black. And we use this as the inoculum because we figured that's where the organisms are. And it served us very well. We could find degraders for all the compounds that we were interested in working with. Okay, so then just to summarize a lot of work that was done by a number of people in our laboratories is that this illustrates the anaerobic degradation of toluene. Okay, remember toluene is a one of these methylated aromatic rings, okay, and it's a component of gasoline. The quantitation is very clear because you can measure what goes into your experiment. You can measure the amount of substrate you put in, the amount of electron acceptor. This is under nitrate denitrifying conditions, so it's not oxygen. And we can measure the amount of cells that were produced as well as the nitrogen that was released from the nitrate. So we can quantitate all these components to describe this reaction that the organisms are taking part in stoichiometrically. It's all balanced, okay? And you can see that this set of reactions is very different. That's all I wanted to say. There's no oxygen that's being added. There's no oxygenase enzymes, obviously, because it's under anoxic conditions. But they go through a number of transformations with these unusual metabolites to a more simple compound that can be further degraded. So this was just one of the examples of the work that Pat Evans did in my lab, but then many other people contributed to our understanding of how these compounds can be degraded. This is an example of the naphthalene, the bicyclic polycyclic aromatic hydrocarbon. And again, through the kind of work that we did, and this is all anaerobic, but under sulfate reducing conditions. So sulfate instead of oxygen was used as the electron acceptor in the oxidation reduction reaction. And we were able to map out very clearly and identify the metabolites which occur as it's being degraded to CO2 eventually, to CO2, which it does because we can monitor the carbon here, we can label the carbon in the compound and find the label in the CO2. So it does happen. It doesn't happen fast, but it does happen. These are slow working organisms. Okay, what did I learn from the environmental engineers during this time of this interest in biodegradation? Well, what's interesting is that the engineers clearly say that more lives have been saved by engineers through chlorination of water supplies than any other public health action. This is just chlorinating water to kill off the pathogens. Mostly it would be readily transmitted pathogens such as cholera and typhoid. For more than 100 years, this has been successful. Wastewater treatment is microbiology, and Professor McCarty actually defined the anaerobic food chain decades before it was described in microbiology textbooks. And he was an engineer because he was interested in what happens in these anaerobic reactors where they use them in waste treatment plants to degrade the waste material and then release compounds and materials that were just non-harmful to the environment. So he studied the anaerobic digesters problems. And I learned to follow the carbon. The term is, follow the money. Well, you follow the carbon because the engineers taught me that it doesn't always matter who the microbe is, it matters what they do. So if you know what's going into a system, into a reactor, and you know what's coming out, then you can figure out, okay, something interesting is going on in here. If we put this very complicated carbon compound in, and we can get CO2 and labeled carbon material out, then clearly there's a degradation going on. So they were never interested in who the microbe was. It mattered what they were doing. And that helped in terms of understanding processes before you could understand the details of how A became B became C. So let me give you a couple of examples. This is a project that occurred at through my collaborations with engineers at Rutgers University. It's the remediation of toluene contaminated groundwater. And so with that knowledge, the knowledge that we know that toluene can be degraded, number one, and the knowledge that toluene degradation can occur in the absence of oxygen, this was a real site that Professor David Paterson and his students worked on. It was a local New Jersey manufacturing site with leakage underneath the building and facilities. There was a leaking underground storage tank. The groundwater was not a drinking water source. And they drilled wells to monitor the samples, the sample area and be able to take samples from that area. And they took the subsurface samples taken in for laboratory treatability studies. Okay, so here we have a manufacturing plant. This underground storage tank leaked, and it turns out it was 95% toluene. So that was actually a very nice substrate for us to work on because we knew the fate of toluene pretty well by then. Okay? We knew that toluene is biodegradable both aerobically and anaerobically. What we did not know is whether this particular site, okay, so here's the water table. This is the unsaturated soil above the water table. It's not known if the endogenous toluene utilizers, natural occurring organisms at the site, were present in this anoxic subsurface and the limiting conditions of what might be helpful in terms of getting this degradation to occur. So this toluene leached, and it could be, and it was a problem. They did not want to dig it up because it was an active manufacturing site. Okay, so the samples were taken into the lab, and they did these treatability studies where they put the material into a column, and they pumped materials through the column for a time period. Nitrate was pumped through one, no nitrate was pumped through the other columns. There were 4 columns, 2 with nitrate, 2 without nitrate. They used the subsurface materials, so they're assuming that there are organisms in this subsurface material, and added nitrate, measured the inputs and the outputs. This is just a reminder of what toluene structurally looks like. So the toluene, the column study results. Toluene was reduced from more than 200 milligrams per liter to 2 milligrams per liter in the laboratory studies. Okay? Residual nitrate was held to less than 0.5 milligrams per liter. We didn't want a lot of nitrate to be still floating around in the groundwater. The theoretical stoichiometry of the toluene to nitrate used, the simple stoichiometry, is a molar ratio of toluene to nitrate is 1 to 6. Okay, 1 to 6 molar ratio of the two substrates going in, and this measured ratio of toluene to nitrate was 1 to almost 5. So the fact that we could quantify a very close ratio of 1 to 6 as 1.5 is evidence that this reaction is taking place. But furthermore, we could do a mass balance on the carbon. We could measure all the carbon going into the system and all the carbon coming out, and we could quantify that 77% was retrieved. So this mass balance also indicates that you couldn't get this amount of CO2 if this toluene had not been degraded. And the mass balance for nitrogen, in terms of nitrogen going in and nitrogen coming out, was more than 100% because we could measure all the nitrate going in and we can measure all the nitrate coming out. So that gave then confidence that indeed this could be an approach for metabolizing this contaminant in the subsurface. So the engineers developed this delivery system, which was these horizontal perforated PVC pipes where the nitrate would be put into and then just distributed into the contamination zone. This is the top view of it. So this is the contamination zone, and all the little dots indicate the monitoring wells. And here are the PVC pipes. This shows the groundwater flow in this direction. And then we measured the outcome. Okay, so if you can see the outcome, I'll try and point it out. This is the level of toluene over time, and the time stamps down here are the numbers of 2-week intervals. Okay, and so toluene was high before any injection of nitrate. Once nitrate was injected, within 2 weeks, the toluene then was reduced significantly. You can see the toluene was high before injection, and then after 2 weeks, it was much lower. To add to the understanding that denitrification was occurring, there was this transient formation of nitrous oxide. Okay. Nitrous oxide is this nitrogen compound which occurs during denitrification by microbes. So nitrate is reduced to nitrite, to nitric oxide, to nitrous oxide, back to nitrogen, releases back. It's part of the nitrogen cycle, releases back to the atmosphere. And this nitrous oxide transient production shows that this was happening. So there was a production of it, and then a further reduction of it to nitrogen gas, which went back into the atmosphere. Okay, so that was convincing. That was quite convincing. And the bulk of the toluene could then be cleaned up without having to dig up, without having to haul away material and then finding another place to deposit it. So there are advantages and disadvantages, just in terms of some general understanding. The advantages of, let's say, using this bioremediation approach is that you get the conversion of the contaminant to innocuous CO2. So it gets converted from the toluene to CO2 and water. So you decontaminated the region. It takes advantage of the naturally occurring microbial community. So we're not introducing any sort of genetically engineered superbug or anything like that. It's generally less costly than physically removing the material or chemical processes, which could be more costly as well as more toxic. And it's accepted. After all these studies, it's accepted by the EPA and other regulatory agencies for certain proven contaminants, such as gasoline. Not all contaminants, but certain proven contaminants. So this is a much more environmentally friendly way to
get rid of the contamination in the subsurface. The major disadvantages that I would point out is that it's slow. You have to wait for the bugs to grow. If you were to dig it up and haul it away, that's relatively quick. You could do that within, several days, but now you have to wait for the bugs to grow. It's a proven technology for specific applications, like leaking underground storage tanks at gas stations, but it's not a proven technology for all contaminants. Okay, so there has to be an understanding of the contaminant. Okay, the other example I want to give you is a site in New Jersey. It's monitoring naphthalene contamination in groundwater. This is a little more in a little more detail, and I hope you can appreciate what we were able to do here. The site history is that this is an old legacy manufactured gas plant, NGP, which was closed for more than 70 years in South Jersey. It's owned by South Jersey Gas. The structures that was on the property and contaminated soil had been removed, but the property was largely owned by South Jersey Gas, and so they were required not to clean it up and haul it away, but they were required to monitor it once or twice a year to see what's happening. So what happened was that they mapped it out very well, and we had the opportunity to take samples at one time, and then 10 years later, we had an opportunity to take samples again to see whether there was any loss or biodegradation occurring. So that was kind of interesting, and that's what I wanted to show you with this. The subsurface contaminant plume was well mapped and monitored regularly as required by New Jersey DEP. The compounds present at the site and in the plume were a whole array: the BTEX, benzene, toluene, xylenes, polycyclic aromatic hydrocarbons, styrene, phthalates, ethers, chlorinated solvents. We just looked at the BTEXs and the pHs. Samples were collected and analyzed two times over a 10-year period. Okay. This is naphthalene. This is data on naphthalene contamination in groundwater, and this was the first time we went there. We just sampled the groundwater. This is the map that shows the groundwater flow. This shows this indicates here the different monitoring wells for this site. And the only thing I would like to point out is, can we answer the question, is biodegradation taking place in this anoxic subsurface? Nothing's going to be actively done, but what is happening in this subsurface? So the samples taken at the monitoring wells, you can see that there was a particular metabolite which we identified. I think this was naphthalene, so it was these metabolites, high at the source of the contamination and then much lower later in the flow, in the groundwater flow, which suggests that there was loss over time. Metabolites formed during anaerobic biodegradation are unique to these pathways, so can be used as vital markers and indicators of degradation taking place. Okay, so 10 years later, we went back, and 10 years later, we had more information too. We had a better molecular understanding of the organisms and the genes and the enzymes that can work in these anaerobic degradation processes. So we went back to take just water samples, and then we filtered the water to take sediment samples that were that had bacteria. Then we took these into the lab to cut it out and to do DNA extractions. And so this was 10 years later. And again, okay, this is the naphthalene pathway, and you can see it's much more complicated because we knew a lot more about it through the work that many other people had done. So these were the metabolites that we were monitoring for. You don't even have to worry about the details. These were the metabolites that we were monitoring for. If we saw these, we knew that degradation was occurring. Okay. And also this BSS4 gene, which encodes for the alpha subunit of the enzyme benzoyl succinate synthase, we understood now after 10 years. And so can we find this gene and can we determine whether there is degradation because of the organism activity and the gene activity? And this is the only enzyme, the benzosuccinate synthase is the only enzyme at that time known to be involved in activation of hydrocarbons under anaerobic conditions. This is the same site, it's just more detailed. Okay? And the plume is here, and the red dots are the wells which we sampled, which were contaminated. The green dots are the wells that we sampled, which were not contaminated. So this was a nice study where we had contaminated sites as well as control sites, so to speak. Wells within the plume, wells outside the plume. Okay, so everything marked with red is inside the plume. Everything marked in green is outside the plume. So I should point out the monitoring included dissolved oxygen, oxidation reduction potential, and pH. So in outside the plume, the oxygen was positive, was relatively higher, 2.5 milligrams, 4.4 milligrams per liter. This one was low. This is an outlier. But inside the plume is consistently much lower, 0.4 milligrams per liter, 0.3 milligrams per liter, which suggests that there's microbial activity. They're pulling down any oxygen that is available. The oxidation reduction potential, okay, a high oxidation reduction potential would normally be seen in more oxygenated samples. So we're talking about 77, 46 millivolts, and 6 millivolts. But the low oxygen or anoxic was minus 170 millivolts, minus 9 millivolts, minus 48 millivolts, indicating it's very anoxic in those samples. And again, we could monitor for the metabolites in naphthalene biodegradation. And all you need to see is that in the contaminated, again, the red is contaminated sites, the green is the uncontaminated wells. You can see that in the contaminated sites, there was a lot more of the metabolites, suggesting that indeed there was a lot more degradation going on. Okay. And then in terms of the biomonitoring, we can monitor PCR, endpoint PCR analysis using 16S RNA gene, which gives us a sort of a total way of monitoring. This is total microbes in our sample. And the red and the green is very clear. The red samples have a much more dense response. The green samples have a much more light response, suggesting that there is much more microorganisms in the red wells because of the substrate and the biodegradation occurring in the contaminated zone. Now, this is specific for the BSSA gene in terms of monitoring for that. And this was really a surprise to us how clearly it indicated this. The red wells showed slight positives, okay? In one of them, we had a very strong positive in the red wells, but the green wells had no bands showing up, which indicated that the BSSA gene, which is specific for the degradation process, we could monitor. We can see in the red wells, the contaminated wells. The qPCR, which gives us more quantitative numbers, indicates that in the green wells, there are, 10 to the 6, so there are about 1 million organisms per mil. But in the contaminated wells, there's 2 orders of magnitude more microbes. So that's 100 times more dense in terms of microorganisms, in terms of being able to quantitate relatively with their the well position. And also we were able to qPCR the fact that we could see a positive signal in the contaminated wells for these for the gene product itself in the contaminated wells, but not in the uncontaminated wells. So it just shows that The wells within the plume were specifically enriched for hydrocarbons. Clearly the organisms in the subsurface was able to take care of doing that and able to mediate the biodegradation. And this just summarizes it. There's multiple lines of evidence which suggests that anaerobic hydrocarbon degradation is occurring in the historically contaminated groundwater. Specific and unique metabolites of the biodegradation pathway are present in the impacted wells. Total bacterial biomass is 2 orders of magnitude higher in the impacted wells, and the specific genes of anaerobes detected in impacted wells, not outside the plume, underscores that their presence is there. There is a presence in the contaminated sites. So these are 2 examples of how having no knowledge a number of years ago to having at least enough knowledge to understand what is happening in our environment and in our subsurface. Okay, so I'm going to end with just a very different sort of ending here. Okay, this is merely to show that our environment is by definition a multidisciplinary one, and to study it should not be confined by the disciplinary boundaries that we find ourselves. Now, this is for the environment because the environment is quite complex, but it's not just the environment. In any subject that you eventually go into, it can be complex enough to appreciate that it's no longer just biology. It's no longer just chemistry. It's all linked together. And that was a lesson that I found being very valuable to me in terms of just my development as a researcher and a scientist, and my development as understanding how we can best understand the environment. These are 3 scientists from 3 well-known scientists and Nobel Prize winners from the last century and 2 centuries ago. Selman Waksman, who's a Rutgers alum, Marie Curie in chemistry, and then of course John Rogan, who built the beautiful Brooklyn Bridge. So there's a microbiologist, a chemist, and an engineer. So one could sort of daydream and see how would they attack the complex environmental problems that we have today with the skills that they had and the knowledge that they had. So I want to end with just a message to the students, and if you let me sort of look at some of my notes because I just wrote this last night. It's a viewpoint from a personal and a professional perspective. When I look out at an audience of students in New Jersey at Rutgers and here also, I'm sort of looking at myself at that age. I'm an immigrant, like many of you might be immigrants. And one of the things that I like most about our state and about teaching at an institution like Rutgers is the rich and abundant diversity of our population here. That just makes it so much more interesting. I came from China when I was 2 years old, and my parents were only going to stay a couple of years and then return, but they decided to stay. So I was not born here, but I grew up here. I grew up in California and New York. And like many of you, I'm an American by all accounts. I sound like an American. I dress like an American. I think like an American. But I'm also different because of my cultural background, right? For example, when I cook, I use chopsticks for everything to cook with. Every pot has chopsticks in it. I drink tea all day, and I like 20th century Chinese art. You might think, okay, we know that America and the US is a country of immigrants, but New Jersey is one of the most diverse states in the Union, and you may or may not know that. We know that there is 50% of the population are considered to be in the minority category in New Jersey. And we know that 1 in 4 New Jerseyites are foreign born. And so this just helps to show how rich and diverse our particular state and this particular environment is. Now, I want to keep this breaking down of borders and barriers as the theme when we think about this. We have crossed then many different borders to come to this country, or your parents or your grandparents. But they're artificial now, especially now, and they don't hold anyone back anymore, because with a keypad and a computer screen, you can go anywhere in the world with just the tap of the keypad, which we couldn't do. Your grandparents couldn't do it, and your parents could, but your grandparents couldn't do that. And also, as a science student, think about this. As a science student, at a young age, we learn that physics and chemistry and biology are separate and compartmentalized fields. But what you realize sooner or later, that all these disciplines are connected. They're not so separate. What is physics? Physics is describes the molecular interaction that occurs when two elements react and form a chemical bond. Physics describes that. Chemistry is what happens when CO2 is converted through photosynthesis to cellular organic material. That's chemistry, and yet photosynthesis is what we call biology, right? These are all connected. The natural world, similarly, and the environment, there are no boundary lines. I'm going to just give one example. Okay, a couple of examples here. The first on the right hand, on the left hand side. This example is several years ago, an explosion at a chemical plant in Harbin, China. There's my pointer. In the city of 4 million people. Okay. So here's the river, the Songhua River in China. The city is somewhere here in this place. The river flows up northward. So, the city, there was this huge industrial explosion, and benzene, a huge amount of benzene was spilled into the river. It's toxic, it's a carcinogen, and they knew about that. And the city officials covered up the accident, but it became a public relations disaster, and also an unwanted international incident, because the benzene-laden river water moved downstream and flowed into Russia. Okay. Water is also a dispute between Turkey and Iraq. Okay. Here's Turkey, here's Iraq, and here's the Tigris and Euphrates River. The headwaters of those rivers are in Turkey. Turkey needs the water because they need the water for agriculture, and they need the electricity that hydroelectric plants can provide. And so they've been damming up the water upstream. Okay. Now, downstream, this is the main source of water for Iraq and also the Syria. So the more that Turkey holds on to, the less water that can go downstream, right? So water is a fundamental human need. So who owns the water? Who owns the water? Water doesn't stop at the borders, okay? Neither does the air. You know, if some of you may remember that coal-fired power plants in the Midwest would release particles. Now they've all been stopped, and they're not as troublesome. But these particles would then float in the air through the normal processes of the atmospheric winds and plumes, would float towards the East Coast, which would be a health risk and caused acid rain, which is now much better controlled. So who owns the air? Okay. What do borders and boundaries mean when it comes to these basic human needs? Who owns them? Clean water and air, they don't stop at the borders, and pollution doesn't stop or respect borders either. So from the point of our natural resources, what we have on this earth is what we'll ever have, and how we treat it or pollute it or preserve it, conserve it, and conserve it can't be limited by borders or by boundaries. So to tie all this together, our studies, when you study whatever science you study, we find that the boundaries of the disciplines are not reflected. For me, the boundaries of the disciplines aren't reflected in the boundaries of our natural world, okay? With this recognition, then, hopefully we can better see and better understand and better appreciate the complexities and then develop better solutions for a better planet. So the last slide I have just shows our world. Okay. When we look at the picture of Earth from space, it embodies the view that we are all traveling through time and the universe together in what is called Spaceship Earth. And this was coined in a speech by Adlai Stevenson way back in 1965, during the time when nuclear war was very much an issue at the UN and a problem. And what he said was very, very memorable. We travel together, passengers on this little spaceship, dependent on its vulnerable reserves of air, water, and soil, all committed for our safety to its security and peace, preserved from annihilation only by the care, the work, and the love we give our fragile craft. So just keep in mind that the resources we have on this earth are the only ones we'll ever have. It's new ones are not being created. That's why there's all this interest in going to the moon or going to the moons of Jupiter or going to Mars for resources that we are depleting. So if my generation sort of started the environmental movement. This was during the 70s and when I was in college and graduate school. Your generation and those following are going to be the ones who continue it, okay? So just keep in mind, borders and boundaries are very difficult to define these days in whatever field you're in. And just keep that in mind and enjoy the rest of your week, okay? Thank you.
So students have another class, feel free to go out and. If anyone has any questions for Dr. Young, of course, we'll have to stick around for a few minutes. Here.
Thank you for coming.